Polymerase exists in every cell of every living thing. It is what's responsible for replicating DNA during cell division. But it will only ever build out a primer in one direction. It's kind of like a one-way street. See, each DNA strand starts with a phosphate molecule. This is called the 5' end. That phosphate is attached to a sugar molecule called deoxyribose, which holds one of the 4 nucleotides. And deoxyribose also connects to the next phosphate, and so this pattern repeats. But the last deoxyribose molecule doesn't have another phosphate connected to it, so it ends with an OH group instead. This is the 3' end, our street exit. The 3' end on one DNA strand is always connected to the 5' end on the other. The second DNA strand always points the other way. And DNA polymerase needs an OH group to attach the next nucleotide, so it always builds on the 3' end. But that wasn't a limitation, it was an advantage, and Mullis knew exactly how to use it. He could create 2 primers that would bind to the DNA before the sickle cell mutation, each on a different strand, pointing at each other. And if he added polymerase along with some spare nucleotides, it would have to extend both primers toward and over the sickle cell mutation. This would create 2 separate DNA segments, both with the mutation. A few miles down the road, he had another eureka. He could just do these 3 steps again. Unzip the DNA, pair the primers, and use polymerase to extend them. He now had 4 DNA sequences with the mutation. And he could make that 8, and then 16, 32, 64, and he realized that if he repeated this chain reaction 30 times, he would have over 1 billion copies of that specific DNA segment, all with the sickle cell mutation. He could cover the entire Earth with identical copies of the license plate. And if he designed different primers, he could change the length of the DNA section that would get replicated. This didn't only work for the sickle cell mutation. As long as you knew the sequence for any part of any DNA, human, plant, bacterial, you could make copies of that exact segment, and the copies would grow exponentially like a nuclear chain reaction. Mullis had invented a DNA photocopier. He named it the polymerase chain reaction, or PCR for short.